So the last couple experiments that you're going to be doing in this lab are going to be with plants. So we will have plant specimens that you will actually be testing to see what happens as they go through osmosis specifically. So the last two are going to be looking at potatoes, and then a water plant, the Elodia, depending on how you say it, are going to be reacting to certain environments. And we're going to be using salt to do this. So as I discussed in the last video, plant cells, well, I should say I discussed the animal cells in the last video about how their plasma membranes can either lyse, which means break, or they can shrivel up and die basically under certain tonic environments. So plants are going to be a little different than that. As we've discussed in previous labs, the cell wall on a plant is going to help keep the shape of the plant cell. So that keeps the cell stronger, and it also makes them a little bit more resilient to changes. So just to review those three tonic environments, right? Remember, if you put a cell in hypertonic environment, that means that there is higher salt outside or solute. We're going to be using salt. So hypertonic has a higher solute outside of the cell. The isotonic will have the same amount of solute both in and out of the cell. And hypotonic means that there is less solute outside of the cell than there is inside of the cell. So this drives the movement of water. Now, just because there's a cell wall there doesn't mean that that affects the movement of water. So the plasma membrane will obviously be affected by water, and then the plant cell can actually lose or gain water through the cell wall as well. So remember that in a hypertonic solution, water will move to the higher concentration of solute outside the cell. For a plant, that is going to mean that the plasma membrane is going to kind of pull away from the cell wall and it's going to shrivel up inside, but the plant is not going to look as much different as an animal cell would. Isotonic, there's going to be movement of water, but no real change either way. And then hypotonic environment, again, the water is going to move to the higher concentration of solute inside of the cell. So there are two phrases here that you should recognize in this lab or learn in this lab. The first is called plasmolysis. So plasmolysis is going to mean that the cell is shriveling up in the plant, but that the plant cell is not going to die as quickly anyway. So plasmolysis means the plasma membrane pulls away from the cell wall, and that makes it look shriveled up in the cell wall. But again, the plant keeps most of its shape. The other is turgid. So when a cell becomes turgid, it basically means that it is filled with water. And again, the plasma membrane is going to push up against the cell wall, but the cell wall is going to keep its shape mostly. It kind of gets a little bloated, right? So turgid, when something is turgid, it's very, very, very firm. So plasmolysis, we have more of like a wiggly cell inside of its cell wall, and turgid is a very firm cell that is inside of its cell wall. So in order to demonstrate this, you're going to do two experiments. The first experiment is going to be using a potato. So you're going to have potato chunks, kind of like mini little French fries, that are going to be put into salt solutions. So one of the things you should know here is that in general, most cells have a 0.9% concentration of salt. No matter what you are testing here, whether it's a potato cell or even a human cell, we have about a concentration of 0.9% salt inside of our cells. So you are going to take the potatoes, and knowing this, you're going to put them into different environments. So the first thing you have to do is dry them off and weigh them. We're going to take them to the nearest gram, and then you're going to put them into three different environments. The first is going to be to put it into pure DI water, what we call deionized water, which is a 0% pure water solution. You're going to put the second potato chunk into a beaker that is filled with 0.9% salt. This is basically a saline solution that like what you would use for your contacts, if you wear contacts, that type of solution. And then the third one is going to be a big one, 5% salt. So you're basically going to be putting it into a pretty thick salt water to see what happens. So you're going to swirl this in the beaker occasionally throughout the course of the time that you are running this, and then you are going to take them out and dry them. So you're not going to be measuring these every 15 minutes or anything the way we did with the bags. You're just going to have a starting weight and then an ending weight. And then using that weight, we are going to determine if this potato lost weight or gained weight, and if that means it was in a hypertonic or a hypotonic solution. So, in your lab book, you will see this equation. At the end of this experiment, you are going to have an initial weight, and then you will have the final weight. So you are going to use these to compare to each other, and you are going to come up with what is called the percent change. So a percent change in weight is just a comparison between the final and initial weight. And then you multiply by 100 to get a percentage. So you take the final weight minus the initial weight, divide that by the initial weight, and multiply it by 100. So then you will end up with a percentage. Now, one of the things to keep in mind here is that you will end up potentially with a negative percentage and a positive percentage, because what that represents is that the potato has either gained or lost weight. So the potatoes that are going to lose the weight will end up with a negative percent change. So their change in weight is negative. This means that that potato was sitting in a hypertonic environment, right? Because remember, when you have a cell that is put into a hypertonic solution, it loses water, which means it's going to lose weight. So then the opposite will be true, right? Potatoes that gain weight will have a positive percent change in weight, and that means that this one was in a hypotonic environment because they, again, if you remember what we were talking about earlier, if you're in a hypotonic environment, then the water is going to move into the cell and it's going to swell that cell up, which means it's going to gain weight, right? We can actually physically measure that. So you're going to do this for your 3 potatoes. Take your initial weight, let it sit for an hour, and then take your final weight. And then the 6 point, so again, through this lab, you have the procedure for the actual experiment, which is 6.4. You're going to fill out the table that is in your lab book. And then 6.5 is going to be the graph. So you are going to be graphing using a bar graph, the change in weight. So you are not going to graph the initial or final weight. You are only going to be graphing the percent change. Now this graph might look a little bit different than other bar graphs you've done because you are going to have a negative number, which means when you graph this, all right, so again, you're going to have a bar for each one of your potato pieces. You had one potato piece in 0% salt, 0.9% salt, and a 5% salt. So you're going to spread those out across your x-axis because this is what you controlled, so this was your independent variable. Then you measured the change in weight, the percent change in weight. So because we're going to have negative and positive numbers, you have to actually put your 0 line here. So a lot of times, and most of you, when you've done bar graphs, 0 is down at the bottom, right? And you just graph up. Well, this case here, we're going to have a negative number. So you have to start at 0, right? So if you have a positive number, you're going to go up and make your graph bar, right? If you have a negative number, right, you're going to go down and you're going to draw that, hit that too hard. All right, you're going to draw that going down. So you are going to have one bar that goes up from 0, one that goes down, and one that should stay about the same, right? You should see an isotonic solution here when you run this experiment as well. So the last experiment you're going to be running in this lab is also using a plant. So depending on your lab, what's happening in your lab, this may be a demo, which means it might be set up for you in the lab so that you can just go look at it. The way that it is written, you're going to make 2 slides. So you can make 2 slides using that water plant. Sometimes we don't have that much of this water plant left by this time in the semester. Sometimes it's not super healthy, so usually your instructor will make it for you so you can actually see it. Otherwise, you have these 2 beautiful pictures in your lab book. These are going to represent a plant that has been put into 100% DI water. So basically we just drop water on it. There's 0% salt. And the other is going to be dripped into, you're going to drop a pretty intense salt solution on it, which is a 30% salt solution. And you are going to look and see what happens to the plasma membranes. So as you can see, as we look at this picture on the left, right, this plant looks about the same as you should remember from your cell lab in lab 3. But the big difference I think you can see, at least with some of these, is that the cell walls have a little bit of pressure on them. They are turgid. These plants are totally filled with water. These were the ones that have the DI water dropped right on top of them. And you can actually see that with the chloroplasts kind of all shoved off to the sides. Now in this picture to the right, this was the one that was put into the 30% salt solution. So this has gone through plasmolysis, as I talked about a little earlier, right? You can see that the cell walls are still intact and pretty firm around the outside. But what you have is the cell plasma membrane that has kind of shriveled up on itself. So all the chloroplasts are actually stuck in here. So you can see all the chloroplasts kind of shoved on top of themselves. This would include all the other organelles as well, right? So everything is kind of grouped into this tiny little plasma membrane. So that is what plasmolysis is, right? We're kind of shriveling up on itself as we look at these plants. So animal cells can't do this, right? Because their cell wall is not present, or they don't have one, not that it's just not present, but animals do not have a cell wall. Their cells are much more susceptible to these environments, right? If you give it pure water, it'll blow up. If you give it salt water, then it'll shrivel up and die. With plants, they have a little bit more flexibility with that, right? They have more structure in the cell wall, so they can react a little bit more calmly, really, to the environment that's around them. And this is very beneficial for them, considering that most plants can't just get up and walk away and find a new environment, right? They are living in the environment that they're kind of stuck in, so they have to be able to adapt to that, right? So this is a major adaptation for plants.